Good evening, everyone, and welcome to the session 8 of Genomic Variant Analysis and Clinical Interpretation. So in this session, I will be talking about the literature attributes in much more details that we will use in ACMG and AMP guidelines for interpretation of sequencing variants. Okay, so let's start with, as I've already told you in the previous session, that ACMG follow 28 standard guidelines which are represented by 28 ACMG attributes. Now, how these ACMG attributes has been named? So, 28 ACMG attribute has been divided into two categories. That is pathogenic, that is denoted by P, benign, denoted by B. These pathogenic variants are further divided into four categories based on the evidence that it is satisfied. If the evidence for the pathogenicity is very strong, it will be denoted with VS. If it is strong, S, moderate, M, supporting, SP. For the benign, for the very strong, it will be denoted by A, strong S, supporting SP. Okay. So now how this attribute has been named. For PVS one, it take P from pathogenic. From very strong, it will take VS. And there's only single guideline, so they will be PVS one. For other, for like example, for strong, if it is S, it will take P from pathogenic, S from strong, and 1 to 4. That means there are 4 guidelines for the strong evidence. So it will be PS1, PS2, PS3, and PS4. Similarly, for the other guidelines, for the moderate, there are 6 guidelines. So it will be from PM1 to PM6. And for supporting, there are 5 guidelines. This is from PP1 to PP5. In this way, the different guidelines and attributes has been named. Okay. So next, how this ACMG attributes had been distributed over different data sets. As in the previous session, I have told you that there are different data sets and methods on which ACMG relies and classify the variant into 5 different categories, whether it will be disease causing or it won't be a disease causing. And these attributes are associated with these data sets and methods. For example, for the population data set, there are 4 ACMG attributes and there are 4 evidence, we can say that, which will be satisfied, like BA1, BS1, BS2, and PM2. Similarly, for computational methods, there will be 2 ACMG attributes or 2 evidence which has to be satisfied by the variant. Okay, and on the basis of it, these 5 categories has been classified for a particular variant. It could be pathogenic, likely pathogenic, VUS, likely benign, or benign. So let's take an example of one of the variant. For example, in here, for RSID 112532, there are 5 attributes. That is PM, which satisfy 5 of the evidence. For the population data set, it is satisfy this PM2. For computational method, it satisfies PP3. For variant type, it will satisfy PM5, and so on. So if we combine all these parameters or your attributes and put it in the ACMG scoring system, it will provide us what type of this variant has been classified. So on combining all these 5 attributes, it will give us this variant is a pathogenic variant. Now how we can retrieve this particular variant? So there are different methods. For the first 4 methods or data sets, that is the population, computational, and variant annotated database and variant type, it could be retrieved from the databases directly. While for the other 4, that is functional methods, segregation data, allelic data sets, and disease association studies, that is GVACI. We have to look at the literature and screen through it to find out whether our RSID will satisfy any of these attributes and can mark it accordingly. Okay. So in this session, I will be talking about these 4 data sets and methods, and will be talking about these attributes in much more details. Okay. So let's start with the functional methods, and we'll use this PS3 and BS3 as an attributes. So if you look at the functional assay, there are 2 attributes which are provided by ACMG, that is PS3 and BS3. PS3 stands for pathogenic strong 3, while BS3 is for benign strong 3. So before going into much detail what are PS3 and BS3, let's get to some of the basic terminology that I use for the functional assay. So if we look at it, for the wild type, healthy, control, and normal. All are assigned to that individual who is not carrier of the variant and not been affected by the disorder. While on the contrary, if there's a mutant, disease, cases, and patient, that will be assigned to the patients that has been affected by the disorders. Okay, so if we take an example of a gene which has a variant, and you hypothesized because of this variant, this particular individual has been affected or the disorder is there. So in that scenario, you will can perform multiple types of assay. That could be in vitro assay or in vivo assay. In vitro assay are done basically in our test tubes to identify or validate the variant of interest, whether that particular variant is really responsible for the disease or not. Whereas in vivo assay, that is done on the mouse model, zebrafish, or the housefly to identify or validate the variant responsible for the disease or not. So you can take multiple samples, you can take samples like patient samples, because the patient samples itself is carrying a variant and can be responsible for the disease. Or you can edit the gene and put your variant of interest and can identify the phenotypic characteristics of that particular variant. Or you can use the plasmid DNA, which already had the variant, and can look at the mRNA or protein expression because of this particular variant. And further, you will do the analysis and compare it with the wild type. So in here, as you can see that when we compare with the wild type mutant 1 and mutant 2, so for mutant 1, the expression, it could be the protein or mRNA expression, has been significantly reduced in comparison to the wild type. While in the mutant 2, there is no significant reduction. It is moreover comparable to the wild type. So if there is a significant reduction, we will mark for that particular variant as the PS3. But if there is no significant reduction, then you will mark it as BS3, that is benign strong 3. So for a single variant, there could not be any single in vitro assay that could validate all the variant. Therefore, different disorder, they would have a different assay for the functional validation. So there's some of the popular assay that has been used for the variant validations. So there are biochemical assay for the quantification of a protein and mRNA levels. So in this, we'll take the samples and isolate the cells of interest, which you think for that particular variant is causing a disease. For example, if it is the immune cells or whether it is the heart cells. And from there, you can isolate the RNA or protein which has been expressed in that particular cells. And you can quantify and look at it, what is the level of this RNA in comparison, there is a comparison between the healthy and the patient cells. On comparison, if there is a significant reduction in the expression of either RNA or protein, then it will be marked as PS3. But if this is comparable, then similarly you will mark it as a PS3. Also, there is one possibility in which this particular patient has a high expression of RNA or protein that also can be responsible for the disorder. In this scenario also, we will mark it as PS3. Okay. Next, if we can take for simulation assay or cellular biological activities, looking at the whether the cells or the protein is functioning properly, even though quantification is perfect, but there could be the possibility they're not functioning properly. So for that, you will perform the simulation assay. So suppose you have a gene, there is one normal gene and one is there is a mutant gene, in which from naive cell, it has been converted to the plasma B cells. This particular gene function is that. So after doing this, if there is a naive B cells, so if there's a naive B cell, if we give the stimulus, like in this case, we are giving a stimulus of a bacteria, it should convert into the stimulate into the plasma B cell, start producing lots of antibody. That should be done in a healthy individual that has a normal gene. But if we hypothesize that this particular variant is causing a disease, not able to stimulate and produce the antibodies, so in the affected individual, the antibody won't be produced. Okay, so if we look at the activity or the immune response or immune measurement from the healthy and the patient, could be a significant difference. You can identify because of this your particular variant, this particular B cells won't be able to produce too much of antibody. In that scenario, you can mark it as PS3, that is pathogenic strong. Also, for different cells, you can't perform a similar assay by giving a stimulation of the bacteria. For a heart cells, you have to give the sodium or ion channels. Or for different cells, there will be different in vitro assay. So different cells have different function and you have to provide different stimulation. There will be different in vitro assay for that. Other than the in vitro assay, you can use the in vivo assay if you wanted to look at the phenotypic characteristics. For example, in here I have taken two embryos and have injected one with the wild type RNA that is normal and other with the mutant RNA that has our variant of interest. Okay, and in after injection, I wanted to observe the phenotype upon injecting the variant, whether what is the phenotype on the heart. And for that, we will perform the cardiac function studies, like we will perform the heartbeat count, and we will look at the whether there is any deformities in the heart or not in comparison to the wild type to the mutant. If there's a deformities and there's a difference in the heartbeat count in between the both of these, then we will know because it's this particular variant, all this malfunction has been happened into the in the zebrafish. Okay, so now we'll take some of the example of the functional assay. Let's take an example of the pathogenic MVK functional assay. There's a study which was done by Huten on the mutation in MVK gene, which are responsible for encoding of malmonate kinase. It causes HITS and hyperperiodic fever syndrome. Okay, so after performing the MVK activity assay for the wild type, which is a healthy individual, it has the activity of 100% in the wild type. But when we look at the mutation which are present in this hyper IgD syndrome patient, that is V377I, I268T, and H20P, they have found out there's a significant reduction. As you can see in this, for V377I, the reduction is more than 60% in comparison to the wild type. While in I268T, the reduction is more than 80%. While for H20P, there is no activity which has been whatsoever seen. So in all the three mutation in MVK gene, there is a significant reduction that has been seen. So for all the three mutation, you can mark it as PS3. While if we also look at another example of benign for RNase H, then you can which are responsible for cardiac goiter syndrome. So in this, there's a study which was performed by John et al. For the RNase H2B and RNase 2C activity assay. So here, as you can see, then the wild type, the activity is 1 by 1 for both of them. But for these three variants, that is the K162T, A1770, or V185G, the reduction in the activity is not that much high. As you can see in all the three variant, it is not even less than 0.8, that is not less than 80%. If the activity is not less than 50% and it's much more comparable to the wild type, you won't consider as pathogenic, it will be benign. So in that scenario, for all the three variant, we will mark it as BS3, that is benign strong 3. Next we'll go through so this much about the functional assay. Next we'll go through the segregation data. Segregation data we will use for the variant has been segregating in a family and causing a disease or not. So for that, there are different attributes that has been used with the functional evidence like PP1, BS4, PS2, PM6, and PP4. For this, we will go for PP1 and BS4. Before going into much detail about the segregation data set of PP1 and BS4, let's try to understand what is Sanger sequencing. In the segregation data set, for the variant validation, it's very important to understand if there's any other member of the family having the same mutation and it's manifesting the same phenotype with the variant. So Sanger sequencing has been considered as a gold standard for variant validation and family screening. So I will go brief about how Sanger sequencing is done. I think in the previous lectures also you have been gone through the Sanger sequencing methodology and the chromatogram. Basically in Sanger sequencing, suppose we have a DNA and we wanted to identify what the sequence of this DNA. So we will perform the PCR with the chain terminating with the help of a ddNTPs. This ddNTPs will share out the DNA and will tag at the end of it with the sequence like cytosine, adenine, thymine. With the help of the capillary gel electrophoresis, this shear DNA will be separated depending on their size. And with the help of the laser beam excitation detection, these different nucleotides will be detected on the basis of what type of peak has been observed. Like in this adenine with the green peak, thymine with the red, and so on. So in this literature screening, when we are doing and for the family segregation and pedigree, this chromatogram will be very handy to understand. So let's go through different types of chromatogram. So as in here also you can see that this is in this chromatogram there are perfect peaks. So there's no double peaks or something like that. So this is the wild type chromatogram. Okay, in this the green will represent adenine, blue represents cytosine, black represent guanine, and red represent thymine. Suppose there is a heterozygous mutation, that is one of the strand has the mutation while the other is the wild type. So in this, at this position, if you can see that there's a two peaks. One is a black peak and other one is a green peak. So in this, the black peak represents a wild type nucleotide, that is G, while there's a green, that there's a mutation on one of the strand due to which it has been converted from G to A. That's why there are two peaks which has been represented here. That is G to A has been done. And these are known as heterozygous single nucleotide variant. Suppose if there's a homozygous variant, so there won't be a two peaks, there will be a single peak. But if you think that the wild type and homozygous will look similar to each other, but when you compare both of them, there here it will be a black peak which represent the G, that is guanine, but here it will be a blue peak which represent cytosine, that is a homozygous single nucleotide variant. There's one more scenario when for the heterozygous indels, that is the insertion and deletion. By looking at the by looking at the chromatogram, you might think that there are multiple mutations at each site after this particular variant, but this is not the scenario. If you can look at this sequence for the wild type strand, there is a C which is present, there is a T which are present here, but in the mutated strand, this particular T has been deleted. So due to the deletion, all the nucleotide has been shifted to one position before. So that's why at this position from T to G, there are two peaks which you can see. One is red and one is black. Similarly, at the second position also, for there will be a two peaks for G and A and so on. So in this way, you can comprehend the different type of indels by chromatogram. Next, we'll go through the segregation data set. So for segregation data set, you will provide for the variant these two attributes, ACMG attribute, that is a PP1 and other one is a VS4, that is a benign strong 4. Suppose there are two mutations. There's one mutation that is a T27R and there's one wild type. In this, there's a both the mother and father are heterozygous and it's an autosomal recessive mode of disorder. Okay, so suppose from here, from mother and father, the child has inherited both the variants or both the defective copy of the gene and it manifested the phenotype. Okay, because there's both the mutation are in homozygous state, while two of the individuals or two of the offspring has inherited one healthy copy and other one is from the mutation. So both of them are heterozygous and one does not inherit any of the defective copy and both of them are the wild type. Similarly, after when they have mated, they have the offspring has inherited both the defective copy and it has a both the homozygous mutation and has showing the phenotype. So in both the scenario, as you can see that it has the mutation as well as the phenotype. So this means that variant is segregating with the disorder and not with the unaffected individual. In unaffected individual, either they will be a carrier, but they won't have the mutation which is in the homozygous state, and they will be marked as PP1. So T27R will be marked as PP1 in this scenario. Other than that, if you look at the other scenario in this, if there's another mutation like A96V and this wild type mutation. So in this, the same scenario in which the mother and father are the heterozygous for the mutation. But if you see at the second generation, this individual is affected, but he has inherited both the wild type, means there's no mutation in this, while the person who is not affected has inherited both the affected copy of the gene. Similarly, when they mated, which are heterozygous, in the affected individual, which is manifesting the phenotype, does not have any of the mutation, while in the unaffected individual has both the mutation, both the defective copy. This represent variant does not segregate with the disorder, because in this scenario, see, both of them are unaffected and has the variant. Okay, but with the unaffected individual, unaffected individual has the variant. So in this scenario, you will mark for A96V as a BS4. Okay, such for PP1 and BS4, you have to mark only when there is a complete penetrance with the disorder, not for the incomplete penetrance or expressivity. Okay, so also for PP1, there are a different scenario, which it could be the PP1 could be supporting, it could have a moderate evidence, as well as could have a strong evidence. PP1 severity depends on the number of individuals, families, and the family generation. So if you look at the PP1, if there's two generation family, it's an autosomal dominant. As in here is, you can see there's autosomal dominant mode of inheritance, and the child has inherited the disorder from the mother, and mother also has that same variant and has been inheriting the disorder. So in such scenario, there is a supporting evidence, because there's not much of the individual which are present having which we can prove as a strong evidence for PP1. But if there's a 3-generation family, and you can see that from mother and father, the child has the mutation that's in a homozygous form, and it also manifests the phenotype. Similarly, for these are heterozygous, and in this 3rd generation also, it has the homozygous mutation and also manifesting the mutation. So this is a 3-generation family, and the variant is segregating with the disorder. So this could be considered as a moderate evidence, and you can provide it as PP1M. Now, if there's a 4 or more generation of the family in which this variant has been segregating with the disorder, and you have identified such individual phenotype with the homozygous variant, in that scenario, you will mark it as PP1 as a strong. Okay. Again, this is the PP1 is done only for the nuclear family. If there's only single family, like this scenario, but PP1, if there's not a 3-generation family, but there are multiple families, like 2 or 3 unrelated family with the variant segregation has been identified. In that scenario also, you can put it as PP1M. For PP1S, if there are families more than 3, in 3 unrelated family, there's a variant segregation with the disorder. In that scenario also, you can put it as PP1S. That's a strong evidence. So let's go through some of the real world example. So as I've already told you, there's one gene that is MVK that is responsible for hyper IgD syndrome. So if we look at it here, there are 10 patients from 6 South Indian family which are suffering from hyper IgD syndrome. And there are 2 mutations which has been found to be inherited in their family. So if you look at the segregation of the variant, so for these 2 individuals which are affected, and both of them have segregated the mutation, and on segregating, they are manifesting the mutation. Similarly, for the second family also, they have inherited the mutation and they are manifesting the phenotype. With the first, second, also for other families also. In this scenario, as you can see that multiple families, there are more than 3 families which are segregating the variant with the disorder. So in such scenario, you will mark V3C7I and this second mutation also as PP1 as a strong attribute. Okay. Now there's an example for BS4. For an Spanish kid, there's one report by LDA for I599T for MEFP mutation. So in this, there's a nuclear Spanish man which is affected with familiar Mediterranean fever with a compound heterozygous mutation. The mother and the father has one mutation. Mother has this heterozygous mutation I591T and the wild type, while the father has M694I and the wild type. And the child has inherited both the defective copies, that is I599T and M694I, and it is affected. So you might be thinking you can put it as a PP1, but if we look at by the Sanger sequencing for its sibling, they both also have the same mutation and both of them are unaffected. So this shows that even though they have the mutation, they does not manifest the phenotype. So both the mutations not responsible for the disease, and in that scenario, the variant is not segregating with the disorder, and you will mark it as BS4. Okay. This much for PP1 and BS4. Next, we'll go for PS2 and PM6 that is associated with the de novo variant. So what is the de novo? De novo is just to represent a new mutation, and there are 2 ACMG attributes which are associated with this, that is PS2 and PM6. What is the de novo mutation? It's basically a new mutation that has arises for the first time in the family due to a genetic alteration in one of the germ cells. That could be the ovum or the sperm cell of one of the parents. Okay. So suppose we have identified one of the mutation in a heterozygous state in a patient, and it's an autosomal dominant disorder. Okay. And it has been confirmed in the families in a mother and father, and all of them we have identified are wild type. That this makes us sure that this particular variant has not been inherited from either of the parents, and it's a new mutation which has been come into the offspring. So if the variant is confirmed by Sanger sequencing or any type of sequencing, then you will provide the ACMG attribute as PS2. That is a very strong evidence. Next, if there is a variant, the similar scenario is there, and if you have not performed the Sanger sequencing or any type of sequence, or you just assumed based on the clinical characteristic of the family history, and you have marked that mother and father does not have the mutation, only the child has the mutation, in that scenario, you will put it as PM6. Okay. So let's go through the example of PS2. So there's one study by Joe et al. For which they have identified a novel mutation, that is E1021K mutation and P110 delta gene, which is associated with the primary B cell immunodeficiency. And they have identified a heterozygous mutation. There's a wild type and there's one mutation, that is E1021K. In this Sanger sequencing, also you can see that 2 peaks. Because of the black and white you can't comprehend, but here if it is written as N, that means it won't be able to find out what type of mutation nucleotide is here, because there are 2 peaks which are present in here. They also performed Sanger sequencing for the mother and father, and they did not identify any of the mutation at this position for father and mother, and both of them have G. So it's a wild type. So with the help of the Sanger sequencing, they confirmed that the mother and father does not have the mutation. And you will put the ACMG attribute for this is as PS2, okay, for E1021K. Next, we will go for assumed, when they have assumed for a PM6, parent has been assumed for, there's a boy with the hemolytic anemia, and they have the mutation of a P327R in band3 protein. They have in this they have performed the Sanger sequencing for the mother as well as the sibling, and they could not identify any of the mutation. But due to the unavailability of the sample of the father, they assume that father also don't have any of the clinic, does not have the variant, and due to its clinical history as well as the family history. So due to the assumption of the parent does not have the variant, this particular ACMG has been marked as PM6, that is P327, on the basis of assumption. Next, we'll go through the another attributes, that is a PP4 for the segregation data set. So the next attribute for the segregation data set is PP4, and in this data set, if the variant of the patient phenotype matches with the distinct clinical feature of the disorder, then you will mark it with the PP4. There should be the four criteria that should be satisfied for PP4 variant attribute. First, that the clinical sensitivity should be very high. That means for the different pathogenic mutation within a gene, there should be a distinct clinical features. Okay. Second, there should be a single gene disorder. And the third, there should be a very less benign variation within a gene. That is, should be less than 50% of the benign variation. And the fourth, the variant segregation should be consistent with the disorder. This should be the four criteria that should be satisfied for marking any variant as PP4. So let's take an example of one of the PP4. In this, there is a study which has been done in our own lab of 10 South Indian patients from 6 families which are affected with hyper IgD syndrome. Okay, so if you look at it in this, there's in this MVK gene which composed of 11 exons and had multiple mutation, has manifested the similar kind of the clinical symptoms. They have a recurrent fever, myalgia, skin rashes, and so on. So the first condition for PP4 has been satisfied. That is, it has a clinical sensitivity is high. The second condition is there should be a single gene disorder. So the MVK gene has caused only one type of a symptom, that is hyper IgD syndrome. The second criteria is also satisfied. The third criteria is that we should have less than 50% of a benign variation in MVK gene. So we have total of 246 variant, out of which there are 70 benign and likely benign variant in MVK gene. So total percentage of benign variation is 28.4%. So this third criteria is also satisfied in MVK gene. And the fourth criteria is the family history, that is the variant segregation within a family should be consistent. So as I have already told you, there are 6 families involved in this study, and the variant, that is V377I, has been segregating in all those families. So the family history is very consistent. So for the variant, that is V377I, should be marked for all the 4 criteria, and we can put PP4 for this one. Okay, so this much is done for the segregation data set. Next is the allelic data set, and there's the 2 ACMG attribute for it, that is PM3 and BP2. So if we look at it here, basically in this allelic data set, it depends on the compound heterozygous genotype. So that depends on autosomal recessive disorder. If it's a compound heterozygous variant, if it is in cis, there are 2 types of a compound heterozygous genotype, cis and trans. If the variant, like in this father, if there are 2 variants within a gene, if they are present on the same copy of the gene, like in this situation, A and B, then this type of compound heterozygous is known as cis. While if this variant is present, suppose capital A present in this copy, while capital P is present in this copy, then it will be known as trans. Okay, so let's take an overview of it. So for example, there is a child who has inherited this particular copy from the father and this copy from the mother. Okay, so this type of inheritance and the genotype will be compound heterozygous in cis. But in the autosomal recessive disorder, both the copy of the gene should be affected for the person to be affected. Okay, so if both the mutation are in the same gene, then this particular gene won't be able to form the protein and won't function properly. But the other copy of the gene is functioning properly, it forming a protein and functioning in a right manner. So the person will be healthy, it won't be suffering from any of the disease. So this is an evidence of the supporting evidence for the benign variation. So we'll mark that variant as BP2. Okay. But suppose there is a condition in which the child has inherited this particular copy from the father and this affected copy from the mother. Okay, so both the gene or both the copy of the gene won't be functioning properly because they have the pathogenic variant in both the copies. Such type of compound heterozygous mutation are known as trans compound heterozygous. In this situation, both the copy of the gene won't be able to make the protein and the person will be affected with the disorder. In such a scenario, you will mark for that particular variant or both the variant as PM3. Okay. So let's go to one of the example. That is the same example as we have gone through earlier for an MVK gene for the 10 families, 6 families and have from 10 patients. So if we look at it here for in this pedigree, both the mother and father are heterozygous for two different variants. For the father, who is heterozygous for a splicing variant, that is C.226 plus 2 del T, while mother is for heterozygous for V377I. When they inherited the mutation, these two affected individuals, both of them inherited the affected copy, means which has the variant, and due to which both of them are affected with the hyper IgD syndrome. Okay, in both the cases. So such type of inheritance is a trans compound heterozygous mutation inheritance. Okay, in this both the copy are not functioning properly and the person is affected. So in this scenario, you can mark the attribute for fourth mutation as PM3. Okay. So this much is for the allelic data set. Next we'll go for the disease association studies. Disease association studies are the genome-wide association studies in which the variant has been associated with the disorder. Okay, so that two ACMG attributes which is used for this, that is PS4 and BP5. Okay, so for the genome-wide association study, there are two cohorts. We take two cohorts. One is the control cohorts, that's the normal individual, and the disease cohort, in which a same type of diseases there in all the individuals. Okay, then we will do the sequencing. Most of them will do the SNP-based microarray, and for this it will has a control specific SNPs, while in this it has a disease specific single nucleotide polymorphism. And we'll compare the single nucleotide polymorphism to discover what type of SNPs are more associated with the disease rather than the controls. Okay. So if you can see that if we look at this particular graph, on the y-axis we have the variant frequency, and on the x-axis are the variant for different cases and controls. So in here, as you can see that the frequency of this particular variant of RSID12345 is highly means a very high frequency in a disease cohort, whereas in the control cohort its frequency is quite low. So this particular variant could be associated with the disease. Now, how will you find out at what frequency that particular variant you could be considered as a disease association? So for that, we have to find out the odds ratio. So how you can calculate the odds ratio, and can for a particular odds ratio you can stabilize for PS4. Okay, so there are various in this case control studies we identify the variant association with the disorder. If the odds ratio is positive, then there's a positive association. That means the variant is responsible for causing a disorder. But if the odds ratio is less than 1, that is a negative association. That means the variant is protecting the disease. Means if you have a variant, this variant is protective for causing you the disease. So suppose let's take an example of one of the variant of RSID 12345, and in which the change of the reference allele is C to T. So in this, we take a hypothetical condition in which we have 100 cases and 100 controls. Okay, for that particular RSID in which there is a change from C to T, there are 50 alleles which have a C, while for T in the cases there are 150 alleles, which will be total of 200 alleles. Similarly, for controls, we have 200 alleles, and for the C, we have 160 alleles, and for T, we have 40 alleles. Now, how you can calculate the odds ratio? So, for calculating the odds ratio of disease, if the allele T is present in comparison to C, we will take the odds of disease in comparison when allele T is present divided by the odds of disease when allele C is present. So in this, what we'll do, so odd of having a disease, we'll take all the allele of T in the cases, that is 150, and divide it with the controls. So 150 divided by 40. Similarly, for odds of disease in the allele C, we will take the allele C of the cases, that is 50, and divide with the control, that is 160. On calculating this, we'll get the odds ratio of 12 of disease with allele T in comparison to the C. This association very high, that is a positive association. That means because of this disorder, because of the variant present in it, it is causing this particular disorder. So on the basis of it, we will decide our ECMG attribute. If the odds ratio is very high, that is greater than 5, with high confidence interval, that is greater than 1, you will provide for that particular variant the ECMG attribute PS4. Let's look at one of the example of PS4. In one of the Iranian Crohn disease patient cohort, we'll look at the not too exonic variant was done by Naderi et al. In this, what they did, they took 120 healthy controls and 90 Crohn disease patient, and they genotyped 21 of the not two variants. On genotyping the not two variants, they identify that 3 of the mutation has very high odds ratio, that is above 5. That is P371T and Q809H has 9.26 of odds ratio, while A794P has odds ratio of 7.71. All of them has a confidence interval of more than 2. So in this situation, for all the 3 variants, you will put the odds ratio who is greater than 1. You will mark for all the 3 as PS4. Okay. So in this way, you can mark for all the literature attributes. So this ends for all the literature attributes, all the functional data, as well as the attributes which we have discussed. So just let's go through one of the variant, how you can mark all these evidences. So let's take one of the variant. So let's take one variant of Dutch mutation, which is very popular. It's known as the Dutch mutation, that is V377I in MVK gene. Now, on the basis of the functional assay, let's start with the functional assay. So if we can see that in this study, which was performed by Houghton et al., in which they identify for a V377I variant is there, then the MVK activity from the wild type has been reduced to more than 60%, as you can see that. So it's significantly reduced. So for this particular variant, we can mark it as PS3. Okay. Next, in the segregation data set, which is our own studies of 6 families and 10 patients, if you can see that there are 3 families in which the variant has been segregated with the disorder. As you can see that the same variant which are present in mother and father has been segregated into the offspring in multiple cases. So there are 3 independent or unrelated families which have the same variant segregation with the disorder. So in that scenario, you will mark it as PP1S. Okay. Next is the allelic data set, which I have told you about the compound heterozygous. If it is trans compound heterozygous and both the copy of the gene is not functioning properly, in that scenario, you will mark it as PM3. So in this pedigree, as you can see that the father is a heterozygous for the splicing mutation, and mother is also a heterozygous for the splicing mutation, that is V377I. And both the child which are affected has inherited in both the copies of the gene both the pathogenic mutation, and they are manifesting the symptoms. So it is in trans compound heterozygous state. So for that, we will put mark for this particular variant as PM3. And the last is the PP4. There were the 4 criteria that need to be fulfilled. And in that, the first criteria, which is the most important, is the patient phenotype should be matched with the distinct clinical features. So in this, for V377I in MVK, matched with the distinct clinical feature, that is recurrent fever, myalgia, and has been matched. So you'll mark it with PP4. So for the functional, for the literature screening for V377I, we have 4 attributes which are satisfied. So on the basis of the scoring system, we'll see in which class it will be falling, whether it will be pathogenic, likely pathogenic, variant of uncertain significance, or benign or likely benign. So if we put it in, if we look at it here, for the literature screening, we are not doing any of the above which we can retrieve from the databases. So according to it, from the functional method, from PS3 or BS3, we are getting PS3. From segregation data set, we are getting PP1, that and strong evidence, and PP4, that is the clinical symptoms are matching, and the allelic data set, that is PM3. We are not getting any disease association study because not been yet done. So we won't get any of the attribute in this situation. So on combining all this and putting it into a Maryland tool, which I have showed you earlier, so it will provide you the pathogenic variant, means classified in the pathogenic variant. So in this way, you can classify a variant into these 5 classes based on your screening of the databases. Also, in the further we will look at how you can do it with the help of the retrieval from the database. And combining all these evidence, you will classify a particular variant. So this is how you will classify a variant by ECMG. We'll give you various variant by which you can learn, and as much as you can practice, so you will know how you can classify these variants. So that much for now, and thank you for listening the video. Bye bye.